Today we are going to discuss few things about muscle contraction. So let's get started. So this is a muscle fiber which is composed of myofibrils, and this is a sarcolemma. As you can see, these are the transverse tubules. These are what? These are the transverse tubules, which are nothing but the inner extensions of the cell membrane. Okay, so and these, what you are seeing in the yellow, this is sarcoplasmic reticulum, and this globular cistern type structure is terminal cisternae of sarcoplasmic reticulum. This terminal cisternae, and this is a transverse tubule. There are two terminal cisternae and one transverse tubule is known as triad of the reticulum. This and this is known as triad of the reticulum. So when the action potential reaches, as we know, the action potential reaches the membrane of the cell. So if it reaches the membrane of the cell, then through the transverse tubules, it moves into the deep of the skeletal muscle. Okay, from the membrane outside, it moves into the deep of the muscle, and then these cisternae, the terminal cisternae, are the stores of calcium ions. Okay, so when the action potential reaches the transverse tubules have, so this is a T tubule, and when the action potential, this is a cell membrane, when reaches the T tubule here, then on the T tubules, this is a DHP receptor. This is dihydropyridine receptors. These are dihydropyridine receptors on the transverse tubules. So when the action potential, these receptors are linked with these receptors. These are the ryanodine receptors. These are the ryanodine R receptors. RYR is RYR receptors, and this is DHP receptor. It is linked with the RYR receptors on the sarcoplasmic reticulum. As you can see, this is the transverse tubules. Here it's DHP receptors, and in the transverse tubules there is RYR receptors. So when this voltage, when the action potential comes, the voltage is sensed by the DHP, that is dihydropyridine receptors, and it then it does a conformational change, which helps to have a change in the RYR receptors on the sarcoplasmic reticulum, which induces the calcium, these are the calcium channels, to open. This was like this, and this opens, and then the calcium in the sarcoplasmic reticulum cisternae, that is the terminal cisternae, okay, the calcium moves out from the stores of the calcium. So till the time the calcium levels are high in the myofibrils, the contraction occurs. How the contraction occurs, we'll discuss later, and like this. And when the contraction is over, the sarcoplasmic reticulum has calcium pumps. These are the calcium pumps which pumps the calcium ions away from the myofibril to the sarcoplasmic reticulum, and there is a protein inside the sarcoplasmic reticulum, that is calsequestrin, which binds with the calcium ions. So the repolarization occurs. Contraction remains till the calcium pump depletes the calcium out in the myofibrils. This calcium pulse remains in skeletal muscles for about 1/20th of a second, and in cardiac muscles for about 1/3 of a second, because of the long duration of the cardiac action potential. So this is a muscle, skeletal muscle, and in the muscle there are a lot of fascicles. Fascicles are the muscle bundles. So in a muscle fascicle there are a lot of muscle fibers, as you can see, and this is one muscle fiber. In it there are a lot of myofibrils, and this myofibril is shown here. In the myofibril there are two main types of proteins, the contractile proteins, and there are bands. Two main filaments are the thick filament and the thin filament. The thick filament is formed from the myosin protein. As you can see, this thick line, this is a myosin one, this is myosin, and this thin line, you can see these are the actin proteins are the thin filaments. Let's study them. This is a myosin molecule which is composed of 6 polypeptide chains. There are 2 heavy chains and there are 4 light chains. These are the light chains, and this is the 2 heavy chains which are wrapped around spirally around each other to form a double helix. These light chains, and this is the head of the myosin. Okay, these light chains help control the function of head during the muscle contraction. So this forms the tail and this forms the head. This is the actin filaments, these one in the pink, and these are the myosin filaments. These are the heads of the myosin filaments, and this is the tail, a little part of the body of the myosin filaments. These are the heads. These form the cross bridges. On the actin filaments, there are some sites, active sites, on which the myosin heads bind. So this is the bridging. This is a cross bridge which is formed while contraction. So a little part of the body of the myosin and this head forms a cross bridge. This arm and the head forms the cross bridge. This is a thin filament, and this is formed of 3 proteins. This is F-actin, tropomyosin, and troponin complex. There are three troponin proteins that form the troponin complex: troponin C, troponin I, and troponin T. The troponin C binds with the calcium ions, and troponin I binds with the actin. Troponin T binds with the tropomyosin. This is a tropomyosin, this dark one, and this light one is F-actin. On the F-actin, there are the black ones, this dotted. These are the active sites, and these active sites are covered by the tropomyosin when the muscle is not contracting. So this troponin complex binds the tropomyosin and actin together with the troponin. So when the troponin C binds with the calcium ions, then it allows the disintegration of tropomyosin from the active sites, rendering the active sites to be free for the cross bridging from the myosin filament. The active sites on the actin filaments are free, and this myosin heads, and these are forming the cross bridges with the active sites. Firstly, it attaches at 90 degree, and then it moves to the 45 degree, moving the actin filament along with it, and that is known as power stroke. The power stroke is what? Firstly, it's 90 degree. Okay, they are attached like this, and then to the 45 degree. So the actin filament move ahead. That is a power stroke. So once the head of the cross bridge tilts, there is release of ATP and phosphate. As you know, these myosin heads have ATPase activity. Okay, now what happens? Now at the site of release of ATP, where it means the ATP is needed by the myosin heads for its disintegration from the actin filaments. If there is no ATP, as in you can say the cadaver, then the cadaveric stiffness will occur. That is, the muscle is in contracted state, it won't relax. To relax a muscle, we need ATP. So when the cross bridge wants to get over, then ATP is used, then ATP and phosphate are released. Then this actin filaments move ahead, and there is new cross bridges. Suppose this was it, and it moves ahead. Now these are the active sites which this myosin head will account. Okay, so it will move ahead like this power stroke will occur, and there will be contraction of the muscle. Then the binding of the new ATP causes detachment of head from the actin. What we are doing like this, we are rendering the actin filaments to move towards each other. So see, this is a relaxed state. These single lines are the actin filaments, and these are the myosin. Okay, so when it's contracted, what happens? The actin filaments move inwards. Okay, because these are the myosin. This will attach and move it inside, as we have discussed. So this is a myofibril, and this is a Z line, and this is also a Z line. Z line is nothing but a protein to which this actin, actin filaments, these are attached. This is a Z line, and this is the actin filaments which is attached to the Z line. So now from one Z line to another Z line, the distance, this is known as sarcomere. Sarcomere is a structural functional unit of a muscle fiber. Okay, so this we can see, this is a Z line, and this is a Z line, this is a Z line, and these are the myosin, these are the actin, and this is another protein known as titin. This titin molecule has a molecular weight of approximately 3 million, which makes it one of the largest protein molecules in the body. It can also come in MCQ. So it's very large, and it holds the myosin and actin filaments. It holds myosin and actin filaments in place. So, and the space between this myofibrils is known as sarcoplasm. The sarcoplasm contains potassium, magnesium, phosphate, and mitochondria, and the mitochondria produces large amount of energy, ATP, that is used in this mechanism. So there are two bands, A band and I band. I band is that area where only, you can see, only actin filaments are there. In the A band, there are myosin filaments as well as little actin filaments. So this is A band is anisotropic, that is it's a dark band, and I band is isotropic, that is light band. And this in the center of the A band, this whole is the A band where there is myosin. So it is center of the A band where only myosin is there. There is no actin molecules. So I'll give you a review. Firstly, what happens? Action potential comes to the nerve endings. Then a neurotransmitter acetylcholine is released. The acetylcholine acts on the acetylcholine gated cation channels, these receptors. And then these gated channels allow large quantities of sodium to move inside interior of the muscle fiber membrane. Here this is a muscle fiber membrane. So the action potential travels along the muscle fiber membrane, then it depolarizes the membrane, and the action potential reaches the membrane, and then through the membrane it reaches the sarcoplasmic reticulum. Here large quantities of calcium ion are released from the reticulum to the myofibrils, and these calcium ions initiate the actin and myosin filaments attractive forces. How? As we have discussed, the troponin C of the thin filament will bind to the calcium ions, and then troponin, which was attached to the tropomyosin, will have a conformational change which will allow the tropomyosin to move, and that tropomyosin, its duty was to cover the active sites of the actin filament. So here it moves and the active sites are open now. On these active sites, the myosin heads will attach, and then that sliding will occur, the power stroke will occur, and it will move these Z lines closer. Once it is contracted, the calcium pump will pump the calcium inside the sarcoplasmic reticulum and allows repolarization and allows the muscle resting or relaxation. So this was about muscle contraction. Thank you.